review some of the results from the survey, including popular imaging techniques, the use of single-cell and spatial technologies, reproducibility, and other challenges in the field. Then we'll move on to the live Q&A. So first up, I'll hand over to Robert for his introduction.
Hello, yeah, thanks, Annie. Hello, everyone. Just to introduce myself, I'm Robert Prevedel. I lead a research group at the EMBL in Heidelberg, Germany, and my group is a mix of physicists, engineers, and biologists, and we are focused on developing new imaging tools and methods, especially in the area of intravital and deep tissue microscopy. And so in the area of neuroscience especially, we have worked a lot in the past in the area of high-speed volumetric imaging in order to sort of visualize the activity of hundreds to thousands of active neurons in worms and mice. And we also brought other techniques such as light field microscopy to biology in order to image large volumes at hundreds of hertz imaging rates. And we've also further worked on methods that allow us now to image deeper into scattering tissues such as the mouse brain. And this we do by exploiting longer wavelength illumination or excitation techniques based on 3-photon microscopy. We also use techniques such as adaptive optics, but also more unconventional approaches that are based on photoacoustics, where we detect sound instead of light, and where we can, in principle, capture an entire brain, which is something similar to MRI, but at a higher resolution. And then last but not least, we also develop tools that allow us to visualize physical properties of biological samples. And for this, we exploit the concept of brilliant light scattering in order to measure biomechanical properties such as stiffness and viscosity in a 3-dimensional way with very high resolution using light. And this is something that other methods in the field of mechanobiology typically fail to achieve. So with this, I hand over to the next presenter.
Yeah. Thanks for that, Robert. And also just to mention that there will be a recording of this discussion that we made available through the same audience link about 1 hour after the session. Yeah. So then, Harry, you're next. Harry? If not, we can move on to Philip. Yeah, you there?
Can you hear me?
Yeah, we can hear you.
Sorry about that. You didn't hear me in the past, right?
No, we can just hear you now.
Okay, sorry about that. So hello everyone, this is Harry Fu from the Ohio State University in the United States. So my lab is particularly interested in studying the molecular mechanism underlying the selective or differential neuronal vulnerability in the Alzheimer's disease and frontotemporal dementia. As shown here on the illustration on the right, and then in many neurodegenerative disease, there are certain neurons and a certain brain region are vulnerable at the early stage of the disease. For example, in Alzheimer's disease, the pyramidal neurons or excited neurons in the nucleus serous, entorhinal cortex, and hippocampus region are particularly vulnerable in the early stage of AD. So my lab is using the 10x chromium single-cell multi-ohm attack plus gene expression, that is a paired single nucleus iron-seq and a single nucleus attack-seq to characterize the cell type specific transcriptomic and epigenomic profiling from the same cells of human AD and controls. We're also using the Visium spatial gene expression combined with the immunostaining on adjacent section to characterize the transcriptomic profiling in the spatial context. The model system we are using is listed on the left. So we're using human AD brain and control brains, and also we're using the cerebral organelle derived from the human iPSCs with the tau mutation. We also have several AD-like mouse models, especially the knock-in mouse models. We hope to use in the multi-omics approach and to study the pathogenesis of AD and the frontotemporal dementia. Thank you.
Great, thanks for that, Harry. And then Philip.
Hello, everyone. My name is Philip Janiak, and I'm a research fellow in the Tom Baden Lab at the University of Sussex, United Kingdom. In the Baden Lab, our research are mainly focused on the color vision. We would like to understand how first the eye, the retina, and after that the brain is processing the visual information. Our main workhorse is a two-photon scanning microscopy, and to do this research efficiently, we are trying to modify the standard two-photon approach just to do the recording much more efficient. As in the biology, nothing is flat and nothing is square. We would like to, using our non-telecentric approach, we would like to modify the shape and the size of our scans according to our experimental needs. So as you can see on the example, we are able to bend our scans to better cover the anatomy of the juvenile zebrafish, or made it recording on the two different planes on two different depth with two different power and different size of the recording plane to do the simultaneous recording on the eye and on the brain of also the zebrafish. Thank you.
Great, thanks, Philip. And then I'll pass over to Miriam.
All right. So hi, everybody. You should all be seeing a video right now, hopefully. So my lab focuses on not only standard MRI, which, as you know, can detect changes in structure, but really what we're focusing on is a new technique called metabolic MRI. So if you haven't heard of this technique, what you can do with this technique is actually you can look at metabolic processes at the cellular level by MRI. So you can inject a probe, such as depicted in the video, pyruvate, and look at the conversion of pyruvate to different metabolites, using an MRI technique. So this is what my lab focuses on. I'm a professor at UCSF, and we've been using this metabolic imaging approach in the context of neurological disorders for the past 6 years. Our main focus is trying to understand how we can apply this new kind of imaging metabolism technique in the context of multiple sclerosis, traumatic brain injury, as well as Alzheimer's disease, with the idea that metabolism is changed and affected and impaired in most neurological disorders. And such a metabolic imaging technique will be very useful to not only detect the disease, but also looking at monitoring of therapeutic response. And so that's it. So this is an example of a few papers that we published on all those topics. Thank you.
Great, thank you. Okay, so for the first section, we'll have a look at some popular imaging techniques. So microscopy, and specifically fluorescence microscopy was the most popular imaging method from the survey. So what are some developments in the technique that have ensured its continued popularity? Robert, I'll come to you first for that one.
Sure. Yeah, I mean, I think as a sort of an introduction, of course, I mean, for instance, microscopy is now really a quintessential tool across all of biology and also neuroscience, because it allows us to visualize with very high optical resolution, so to the diffraction limit, right? So below a single micrometer, that allows us to resolve, cellular events at high spatial, but also temporal resolution, and also to have molecular specificity, which of course comes from the development of fluorescence labeling technique, fluorescent proteins, labeling techniques, probes and sensors that can report in the area of neuroscience, for example, about neural activity, but also on other neurotransmitters such as dopamine or glutamate. And I think in terms of recent developments, of course, labs such as ours, but also others and companies have always tried to push the performance of microscopes in terms of speed and resolution and volume areas. And now we can record from thousands of neurons in awake mice, for example, but also other model organisms in neuroscience. We can resolve brain structure at very high resolution, even in deeper brain regions, subcortical brain regions. And what I find quite exciting now is that one can do all of this, for example, also even in freely behaving mice. So there's the field of sort of miniscopes, both 1 photon and also 2 photon miniscopes that now allow us to look at brain activity using optical tools in freely behaving mice. So that would be my answer to your question, but I'm sure others can also chime in.
Great, thanks for that. And Harry, have you got anything that you'd like to add?
So I think besides the Rob mentioned about the 3D and the deep tissue imaging at the cellular resolution, and then also several companies have developed like a SIMSTAT and a SOPHIE or STORM, those kind of high super resolution microscopy. I just want to add one on is that like expansion microscopy is developed by Boyden and the colleagues from MIT, and then they're using the method to enhance the microscopy resolution by swirling a sample rather than using the high resolution equipment. This may be helpful for some small lab cannot afford, those super resolution equipment. Thank you.
Great, thank you. And so MRI and fMRI were also popular techniques, and similarly, have there been any recent developments in these methods? So Miriam, I'll come to you for that one.
Yes, of course. Yes, so I'll start with just MRI. As you know, the advantage of MRI is the fact that it's non-invasive and it's translational. I think some interesting developments in MRI in the field have been the use of low-field MRIs that can be used everywhere in many places and allow actually still to have a pretty high resolution of the human brain. So trying to fight health disparities in that sense. If you go more into the research side, high-field MRI is also a huge development with now reaching high resolution at 7 Tesla. So most clinical systems are 3T right now, but we have a lot more 7 Tesla, even an 11.7 human system that was installed in France last year. So allowing for really exquisite resolution at the micron level of MRI. As of fMRI, so as you know, fMRI looks at the brain function by looking at changes in oxygenation. I think it's used a lot in the cognitive field, but right now what we're seeing in the clinic is what I was talking about, is fMRI is kind of being not replaced, but a little bit by metabolic MRI. I think by looking at metabolism directly using metabolic imaging techniques, we have a lot more information than by looking at brain oxygenation. So those metabolic imaging techniques are being developed all across the world right now. The more fancy name is it's hyperpolarized carbon MRI, if you ever come across it. It was created 10 years ago, discovered 10 years ago, and 15 years ago, sorry. And now it's in 25 centers across the world, and probably 70 in the next 2 years. So it's kind of a new MRI technique that you might see coming up around you pretty soon. So that's one I'm very excited about, of course.
Great, thank you. And then, so personally, which imaging technique excites you most at the moment? So Philip, I'll come to you first for that one.
I mute myself. Two-photon scanning microscopy. This is something what I'm working on, and this is something what is the most exciting for me. I can still see that a lot of the possible application for the 2-photon and 3-photon scanning, and still there's a lot of things to improve in this field, and I'm extremely happy that the people are working very hard, like Robert, to make this technique better and better.
Great, thank you. And then Robert?
Yeah, sure. Yeah, I mean, to add to what Philip said, I think I fully agree. There's a lot of 2-photon or multi-photon techniques at the moment in the area of optical neuroimaging. I think, like Philip, pushing the amount of volumes that we can record from. There's a lot of techniques now out from different labs. We can now image almost the whole cortical region in the mouse, maybe even at the same time or at different cortical regions to look at how they sort of are involved in processing. I already mentioned the area of miniscopes, right? Which are head wearable microscopes, so mice can freely move around. I'm very excited about that. And then I think last but not least, what I'm truly excited is that, I mean, most often with optical microscopy, we look at sort of indirect readouts of activity. So like in fMRI, you know, where we look at brain function through oxygenation or metabolism, as we just heard from Miriam. In fluorescence brain imaging, we use, for example, calcium sensors, but these are only proxies of neural activity. And I think now there's been a lot of development in the area of voltage indicators or voltage probes that can report on action potentials, you know, which are sort of the digital bit of the information processing in the brain. And now we can also start to record this optically. And for this, we also need to now increase the performance of optical microscopy by a lot, because the temporal resolution of action potentials is much faster than what we can get from calcium indicators. So that would be my answer. Thanks.
Great. And then Harry, which imaging technique excites you most at the moment?
So personally, I think the expansion microscopy combined with the super resolution microscopy excites me most at the moment. So then with that, we can perform like a spatially precise in situ RNA sequencing, and also can easily map the image of the synapse, cells, and neural circuits. So that's my prefer. Thank you.
Yeah. And then finally, Miriam, which imaging technique excites you most at the moment?
So I'm going to be the odd duck that doesn't talk about microscopy. I think for me, on the MRI side, what excites me the most is those metabolic imaging techniques. I think right now there's a lot of clinical need for many patient populations that are treated with different drugs that don't work for them, and sometimes for months at a time, including cancer patients and MS patients. And the MRI readout that we have, as we know, is non-specific. So patients can be treated for months without knowing if the treatment is good for them, which is means that they have poor quality of life, and it also costs the healthcare a lot. So what I'm excited about is really going into the realm of personalized medicine for those patients and being able to adjust a treatment for each patient using this metabolic imaging readout. So looking at metabolism by MRI, using a conventional MRI machine will allow us to really see if the treatment is working for each patient and adjust the treatment accordingly. So improving the quality of life of those patients and making sure we give them the best treatment possible. So that's on the translation side what excites me the most.
Great, thank you. So moving on, our next section is on single cell and spatial technologies. So they've continued to grow year on year since they were developed. So in our survey for both single cell and spatial, we found that the main barrier to incorporating them in research was not knowing enough about them. So where can people begin with this? So Harry, you're our single cell and spatial expert, so I'll come to you for this.
Sure. So as we know, like the function of many biological systems, such as the central nervous system, and then particularly the human brain, depend on the spatial organization of their cells. Those cells are highly heterogeneous. So in the past decade, the highest throughput techniques such as the single cell, single nucleus RNA-seq, ATAC-seq, or spatially resolved transcriptomics have been developed to quantify those gene expression in single cells or single nucleus and in space. So if you know very little about those techniques such as the 10x genomics, single cell, RNA-seq, multi-omic, or Visium platform, so you can visit their website. They have plenty of very useful information about the products, applications, tutorials, and webinars. Or you can contact them. They are very nice, friendly, and then they can answer your questions, help you out. And then question you may ask yourself first is that, what's your sample? Is it live cells or fixed cells or frozen brain or fixed brain tissue? And then you want to perform single cell or you want to using single nucleus for the gene expression assay? And are you interested in particular cell type like astrocyte microglia, or interested in general all the cell types? Also, what kind of section have you? Have FFE section or frozen sections? And then they have different things, right? And then also, lastly, you have to think about what the scientific question you want to address. You want to do just like a screening purpose, or you just try to do target validated. Then you can decide you use antibodies like a spatial result transcriptomics at sports level, or close to single cell level, or you want to using high resolution targeting sequencing at the cellular or subcellular level. So those things I think you can start with, in my opinion. Thank you.
And then another challenge that came up was that the technical barrier for these experiments is quite high. So do you have any tips for carrying out these studies and experiments?
Yes, these experiments are a little bit challenging, especially for new users. So my tip is that before the real experiment, always practice a few times. And then, for example, you can practice how to prepare the single cell or single nucleus preparation, and then to make sure the viability of the cells or the quality of your nuclei are good enough for the gene expression assay. As for the Visium platform, and then without the help of the recent developer like a site assist instrument, and then you have to practice how to mount those sections on the little area on the gene expression side. So that's a little bit challenging for new users. And then for the gene expression assay, when you come to that, just read the protocol carefully and then exactly follow the protocol, understand each step. And if you really have some difficulty, and then nowadays they have some core facility or service provider, they can do those jobs for you. And then, but you have to make sure your samples are in good shape or quality. In my opinion, the most challenging part is the cell segmentation and how to integrate multi-omics data, integrate a single nucleus RNA-seq, single nucleus ATAC-seq, and a spatial transcriptomics data set, and that's a little bit challenging. And then in our case, we think that using the sample from the same patient or same animal will be helpful for the integration. And although the downstream analysis, you need to discuss with the bioinformatics group, and then to make sure the data are meaningful in biology. And lastly, always validate the interesting gene and with the method like a single molecule fish assay, qPCR, and the immunofluorescence, and then before you do the complicated and expensive biological experiment. That were my tips. Thank you.
Great, thanks for that, Harry. So our next section is on reproducibility. So reproducibility poses an issue to many areas of science, and in our survey, 94% of people agreed that reproducibility is an issue in the field of neuroimaging. So firstly, do you agree? And secondly, how do we go about tackling it? So Robert, I'll come to you for that one first.
Sure, I try to provide some answers. I mean, of course, the question about reproducibility is a big one and a large one. And, very often the question of reproducibility is probably on the entire study, right? That does not only involve the microscopy parts. And so I'm not probably the expert to talk about this, but of course, all the data that is involved in neuroimaging studies should be openly shared. I mean, of course, to a large extent, it's image data, but image data is also only useful if there's sufficient metadata with it, that give all the parameters on how the images were acquired. And then, of course, I think there needs to be more of a push to also share analysis and analysis pipelines, so that other people can actually go from the data from the raw imaging data to the analysis and come hopefully to the same conclusions. Then in the area of the microscopy itself, and we build custom microscopes, of course, reproducing those in other labs can be, of course, very challenging. I mean, things that our community does and also we do is to, of course, sort of share custom engineering files, like cut files, that allow particular pieces to be manufactured also in other places. We also share custom code, so for example, control our microscope to analyze parts of it. And I mean, for some of our projects, we even have maintained small websites that have a little bit of a step-by-step protocol of how to assemble, for example, a light field microscope. So that's as much as I can say to that topic, but I'm sure others can add more.
Yeah, great, thank you for that. And Harry, did you have anything that you wanted to add to that?
First, yes, I agree. Yeah, so my short answer, I think if we increase the sample size, not always have a proper positive and negative control for your experiment, and also, like Rob mentioned, have detailed protocol and codes and shared, and then I think that will increase the reproducibility. Thank you.
And then, Philip, do you have anything that you wanted to add to that?
Yes, first I agree with the speakers, and the point, the two issues. First, education. Being a physicist, it's easy to understand the idea of the experiment of microscopy, but a lot of people working in neuroscience doesn't understand that. So the education is important. People are doing some sorts of affiliation. We are talking, for example, about the podcast measurement, which is absolutely underestimated. What we are measuring in the reality is change in the instance, which is somehow correlated with the activity of the calcium, which is again correlated with the neural activity. So understanding of what your experiment is about is I think the key point. And the second issue, the possibility is I think is the max price of the experimental. A lot of people are using shell facilities like core facilities, which is absolutely fine, but it's difficult to optimize my experiment to simply make it more efficient and try to produce the genome models if you can define specific setup. And after you someone else change the settings, etc. So if the equipment can be cheaper much more affordable, the labs they can have their own setups in the reproducibility of their presence.
Great, thank you. So, as just mentioned, sharing imaging data could help with the issue, but our survey found that just 18% of people openly share their imaging data. So 29% of people were afraid that others would publish results on their data, and 24% said that they don't know how or where to share it. So how and why should we encourage more people to share their data? Miriam, I know you recently published a paper on this traumatic brain injury.
Yes, that's correct. Yes, so like all those issues that we mentioned earlier about online reproducibility and sharing data, it's almost impossible to assess how reproducible an experiment is and even the cost. And sometimes that it takes you cannot do it in your lab 1,200 subjects, 500 mice. It would take a long time. There's also some interesting variability in looking at, for example, traumatic brain injury. The models are not and if you have traumatic brain injury in a mouse, it's not going to be the same from one mouse to another. So there's intricacy, variability, and complexity when you start looking at disease model. So sharing the data and knowing all the data together from multiple studies, like we did in the paper. The paper is very simple. It's an open data commons from traumatic brain injury mouse data. So if you have done any mouse traumatic brain injury mouse and you've done microscopy or MRI or other type of techniques, you can visit it to this open data commons. And then all of a sudden, if everyone does that, you have we have 1,200 data sets that have all parameters that are very complex data sets that you can start putting data from. So as using artificial intelligence approaches or multidimensional analysis, and really taking advantage of everyone's contribution to the field. It doesn't mean that you cannot publish your data, like you should publish your data once you have them, but even once it's published, finding such a repository to put your data so that others can do some data mining, like in the clinical setting, for example. There are a lot of places where you can put your MRI data, your brain data, and then you have people that are experts in AI that are trying to find patterns and things and push the field of analysis forward. So encouraging, I really encourage people to share their data and to find repository and platforms. I also think that scientific journals should encourage publication of such data sets, like we did in the Open Commons for TBI. If someone is wanting to put data and put a bunch of authors together to publish a new data set, it should be, I think, also supported by journals as something very positive and published. So, yeah, that would be my view on that.
Great, thank you. And so, moving on to other challenges, we asked our audience what they thought the greatest challenge in the field was. So in the survey, 43% of people said accessibility to specialised tools was their biggest challenge, and others commented on the cost of equipment. So how can we make specialised tools more accessible? So Philip, you've already talked a little bit about customization, but could you talk a bit more about that and the open hardware movement?
Yes, this is one of the most important approaches in our lab. Also, Robert was mentioning that he's doing the same in his lab. So just publishing everything what we are doing as an open repository. In the field of neuroscience, there's a lot of communities like Open Neuroscience or the GOSH, the Gathering for Open Science Hardware communities who are working together to try to publish all of the hardware solutions which they are producing in the different labs in the world. The main reason why people are doing this is just to make it much more accessible for the others. Because of course the companies can do, for example, a simulator for us, but the companies are not interested in building something that will be used only by the 3, 4, maybe 5 labs in the world. So we need to do this by ourselves, and to give possibility to the other people to reproduce it. We also need to design it in the manner that it will be easy to rebuild this setup. So we need to prepare the proper documentation, and also design it in the way that it will be easy to use off-the-shelf optical elements, for example, and 3D printing as I think the main method which help us to build a design like a lot of the elements for our setups.
Great. And Robert, did you have anything that you wanted to add to that?
Yes, sure. I mean, in addition to what Philip just said, of course, what we try to do or what the community has done is, for example, to sort of design simple versions of some of the microscopes. I mean, in the area of, let's say, light sheet microscopy, this has been done with the open SPIM or even flamingo concept, where then these can be shipped around to different labs that need it. But also, of course, we need to increase our efforts to actually get sort of our prototypes that we sort of build in our labs somehow out there for the users. And I mean, there are more and more so-called translation centers that are appearing worldwide. I mean, here at EMBL in Heidelberg, we just opened the big imaging center, which actually will allow some of our microscopes, but also other state-of-the-art microscopes to be opened up to the research community, where then everybody can come and use them. And similar efforts are underway, I know, also in Genelia or Rockefeller. So I think we have to work on all these fronts in order to really actively try to disseminate the technologies that we build, because without that, they will not have the impact that they can have. Thanks.
Great. So the second biggest challenge from the survey was imaging analysis and image analysis. So do you have any advice for selecting correct image analysis approaches? Harry, I'll come to you for that one first.
Sure. So I mean, depending on the research question and on how big of your image files. So for example, in our study, and then the human brain tissue is usually a bit big. And then when we took the tile images, they are usually like 10 to 20 gigabyte. For example, we do the ion scope, the high-plex assay, and then we need to register a few rounds of the staining of those big image files. So in our hands, we think we're using most time the ImageJ or Imaris or some like sort of tools in the GitHub, like Wizrag, is work well in our hands. Yeah, that's my answer.
Great. And then, Robert, did you have anything that you wanted to add to that?
Yeah, sure, I can try. I mean, in addition to what Harry mentioned in terms of tools like ImageJ, right, which is very popular, I think, of course, that the question is always like, does a specific tool, let's say for calcium image analysis also for already exists? A lot of, I guess, novice users don't really know. And that is, of course, then the key question: how can I find out what the correct or best analysis approach and tool is? And I mean, for this, I can only recommend two things. A is to maybe talk to local experts and resources. I mean, big institutes like also EMBL, we have dedicated staff that is trained as image analysis and that try to provide starting help for some of these questions. There's also a great forum, the forum.image.sc, where people can post questions about image analysis, and then they will hopefully be helped or at least put into the right direction. So, of course, as a community, we also realize if we generate a lot of images, we have to also analyze that. And I think a lot of universities and research centers now realize that, they need specialized support staff and in their facilities and on their sites. And I hope that this will continue, that there will be more expert help for people who want to get into image analysis. Thanks.
Okay, great. And then, so what do you think the greatest challenge in the field is and how can we overcome it? So Miriam, I'll come to you for that one.
Well, the field of neuroimaging is pretty broad, but I think what we just touched upon is very key. The reproducibility in data sharing for me is very, very important. I think we have to work as a community, even for, I guess, image analysis. A lot of challenges are really data sharing and the fact that we need to publish and people want to publish and they are scared of sharing that data. And they are also worried about the analysis. When I read a paper, you need to have a lot of details on the analysis to make sure you understand how it's done. So I think what we're trying to do, at least in the field of metabolic MRI, is, and a lot of other fields are doing it too, is working in groups and doing consensus papers. So as some is a way to overcome such issues. So getting together with a bunch of experts in the field and trying to see what's the best. I don't think we can do a correct way, but the best way that we can process data or the best way that we can share data with each other. So working in groups, collaborating, and not being afraid to collaborate and working in consensus group if you can. I think it's very crucial, and it will change those issues in the field. Hopefully, overcome them.
Great, thank you. So then, in our last section before the Q&A, so remember to submit any questions that you want to pose to our experts in the Q&A. Let's look towards the future. So what are you most excited about in the field? So excited applications of these techniques, that kind of thing. So Robert, I'll come to you first.
Sure. I think I already shared some of the exciting directions in the field in one of the first questions, but maybe to just repeat, the ability to use optical techniques to really record neural function, especially, but also structure over large volumes, in entire brains. This is certainly exciting. The ability to do this also in freely behaving animals, and then to do it at a time resolution where we can actually look at voltage, which is sort of I think the most important communication that is being used in the the brain. But then what I'm also truly excited about in the future is that I believe, the microscopes will become what we call a lot smarter, in a way that we can sort of record activity, we can then also analyze it very fast, and thereby sort of maybe also change stimuli that we give to animals such as the mouse, in terms of, let's say, virtual reality or so. And in this era of smart microscopy, of course, artificial intelligence plays a super important role. And so I think we're going to see a lot of new things happening on this interface between, really hardware and instrumentation development, but also the use of artificial intelligence and, what I call smart microscopy. So that's what I'm excited about. But I'm sure others have also other opinions.
Okay, so Harry, what are you most excited about in the field?
So personally, I'm most excited about the high speed and the high throughput and the high resolution imaging of the level or the location of the DNA, RNA, and protein in the brain tissue at the single cell or subcellular level. So these kind of tools can really open a new era of the neuroimaging and the sequencing, and help us understand the health and the disease at the cellular, subcellular, and the molecular level. So that will be my most excited about. Thank you.
Great. And then Philip?
I think for me the most exciting is a combination of the imaging and optogenetic stimulation, which can allow us to not only look at the brain, but also to control the function of the brain. So a lot of groups are now working on stimulating the population and the signal of neurons with a very, very high spatial and temporal control of it. So this is the one thing. And the second is the development of the light sources which we are using for the imaging, that we don't need to anymore use those extremely expensive and big laser sources, for example, for the multi-photon imaging. But now it's the fixed wavelength lasers are much cheaper, much smaller, so it could just push the entire field forward.
Great. And then finally, Miriam, what are you most excited about in the field?
So besides the impact of metabolic imaging, as I mentioned before, on patient population, what I'm very excited about is also the progress that is being made in all those imaging fields that we mentioned. So and how we can also combine the different imaging modalities. I think it's pretty key. So, for example, we just did a study that is submitted, not published yet, in which we combined metabolic MRI with spatial genomics. So I think if imaging modalities and different imaging modalities can feed each other, spatial genomics has the specificity and information that, of course, we're lacking by MRI, but it can help us understand what we're seeing in metabolic MRI in mice, and then in patients in the future. So the complementarity between all those fields and how we can learn from each other and combine this in larger studies, very exciting for me.
Okay, great. So thanks to our expert panelists for that insightful discussions. If you do have any questions for our panelists, then please continue to submit them using the Q&A tool to the right-hand side of your screen. So we've received a number of questions so far, so we'll address some of these now. So this is a question that came up from our survey. So do you have any good software to connect single cell and spatial sequencing data? So that one's for Harry.
So sure, you guys noticed there's a recent Nature Method paper, and then they compared like a 16 integration method, and then they found like a Tangram and then GMVI or SPA GE, and then those perform very well for predicting the spatial distribution of your RNA transcripts, while the cell-to location and then spatial DWLS or RCTD method, those tools are very good for cell type deconvolution of the spots in your histological samples. So I think in our lab, we're using the cell-to location for cell type deconvolution, so it worked well. And then I think that's maybe a good tool if you want to do cell type deconvolution. Although there's it's a very challenging field for the integration of different data set, and then especially single nucleus versus integrated with the spatial data. So many groups are still trying to figure out exactly how to integrate them in a correct way, and then also get more information from those two data sets. That's my answer. Thank you.
Great, thanks for that, Harry. So another question that came up, what do you think the future of AI is in the field? How can we make it more accessible? So Robert, I know you already, you mentioned that slightly in your previous answer, but I don't know if you wanted to have a go at that.
Yeah, sure. Maybe very briefly. I mean, as I mentioned, I think this will be very powerful, right? Because the combination of, let's say, very fast image analysis together with, microscopy hardwares can allow us to sort of react, in real time to sort of the neural function, for example, that we measure and record. And, we have now all these optical modules and tools, if you look at the literature that allows us to record neural function on different spatial scales and temporal scales, but also to perturb them, right, with optogenetic tools and means. And, I think sort of all this together will become super important in the future. So I think the future is bright for that. And the question about how to make this all more accessible sort of also goes back to what we were discussing beforehand, right, that we just need to be very careful and not careful, that we really need to ensure that we also sharing the tools, and all of this can be done in principle, but then of course really ensuring that it can also be reproduced in other labs. That's a top priority. Thank you.
Great, thanks. And then, so this is one that's been submitted for Miriam. So could you specifically describe how metabolic MRI has been validated? So the person said, I do not understand how you can trace single metabolite.
Yeah, I didn't go into the details of the technology, but so the technology you probably heard of MRI. MRI can detect water, but MR spectroscopy is another technique that has been around for a long time, in which they can detect metabolites by looking at their spectral signatures. You get peaks. And proton spectroscopy has been used in the clinic for the longest time. With this technology, what we're doing is we're taking a C13 label metabolites, like that is endogenous to our body, such as glucose or pyruvate, that is non-radioactive, and not like that. And you're putting this metabolite in a system called a polarizer that makes it hyper visible by MRI. So it's a quantum reaction that happens, and you make this metabolites like a really high visible contrast agent that you can inject then in a human or mouse or cells. And then by looking at the signature of those metabolites, each metabolite has a different peak, a different ppm frequency, resonance frequency. You can see the metabolites being converted into subsequent ones. So you can look at pyruvate being converted to lactate, or to bicarbonate entering the TCA cycle. And there are many probes that you can polarize and inject that way to specifically look at different metabolic pathway. And this technique have been discovered 15 years ago and validated now. There are more than 400 papers using this technology in preclinical models. And we have now 22 clinical trials going on across the world. So validating the technique of looking at those different pathways. So I'm happy to share papers and reviews if that's of interest as well. I hope that answers the question.
Great, thanks for that. And then Harry, this one's for you. So could you describe how single cell imaging has been validated?
So though I'm not sure exactly I understand this question. So to me, they maybe want to know some like a single cell level in sequencing compared with the, like a current like a Visium platform, and then the spots that the diameter of the spot is a 55 micron, so they cover multiple cells, not just a one single cell. So recently, and then like Tanks Genomics, they designed the Gazillion platform, and then that can go to the single cell level, and then even below, like subcellular level, can go to, I believe it's a 100 nanometer level. And then that really can answer, the RNA transcript captured within that cell, or even the subcellular organelle level. Instead of from multiple cell type, you have to think about using other software and then to predict the cell type in those spots. And then for validation, I think as I mentioned in the past, and then like IronScope assay is a single molecule fluorescent in situ hybridization assay. It really can, robustly validate your single cell imaging data. Thank you.
Great, thank you. And then this one again is a bit specific. So will imaging be able to give us insights into the possibility of axonal injury in concussions? So I'm not sure if anyone wants to volunteer to take that one.
Maybe I can try just briefly. I mean, of course, the question is in which model organism or in which organism, concussions. I mean, if you go about the human brain, I think only MRI sort of can have sort of the capability to image at that level. Axons are, however, subcellular in their size. I mean, they are on the order of, let's say, a micron or so, 1-2 microns maybe at most. So you need high spatial resolution. I think in sort of more transparent model organisms, also in the mouse, depending on very superficial cortical layers, imaging does have the resolution to look at axons. How to detect injury, I think that would then also depend. I'm not really the expert on the biology of all of this, but there maybe someone else of the panelists can chime in.
I can chime in quickly on this if that's okay. I mean, the with if it's in humans and even in mice, as Robert said, that it's really hard to just really look at axonal injury and to be that specific. I mean, even with metabolic imaging, there's no probes or metabolic or contrast agent that we can inject that is going to be only processed in axon or give us that specificity. Maybe PET imaging actually would be one of the only as one that would give us some sort of specificity there. But I think it will give us insights, yes, but being specific to axonal injury, likely not, at least by MRI and metabolic imaging. But in larger systems like mice and humans.
Great, thank you. So we've probably got time for one last quick question. So this one came up in the survey. So for image analysis, what are the benefits of using analysis pipelines that, analysis that's integrated into your equipment versus exporting the data and analyzing it elsewhere?
So maybe I will say something about it. I think, again, everything depends on the experiment which you are doing, on the animal model, on the questions which you would like to ask. Of course, a lot of the software which are controlling the equipment are giving you the possibility to make some sort of analyze or pre-analyze. But for example, to share the data, the greatest solution is to try to save them just simply in some sort of open source format. And try to analyze them using the methods which the other people are also using. So using the software which is available to analyze. But again, everything depends on the experiment, on the animal model, on the questions which you like to ask.
Great, thank you for that. So I think that's probably all we have time for today. So thank you to our panelists, Robert, Harry, Philip and Miriam, for their time and insightful responses. And you'll be able to access an on-demand recording of today's event using the same link that was sent to you via email, and we'll also upload a version to www.biotechniques.com/webinars. So I hope you've all enjoyed today's panel discussion. Thanks very much for tuning in, and I hope to see you at one of our upcoming BioTechniques webinars. So bye for now.
Bye.
Bye. Thank you.
Bye.